I'm here at the Hub Culture Pavilion in Davos. The moon is starting to rise, just as Paula Hammond has come to join me. Thanks very much for coming along.
I'm having a lot of fun, thank you.
Now, you're the head of the Department for Chemical Engineering at MIT.
That's right.
And also the Koch Institute for Integrative Cancer Research.
That's right.
Now, tell me what you're here telling Davos about.
What I'm telling Davos about is the way we can use engineering of nanomaterials to address cancer. And I'm in this Koch Institute for Integrative Cancer Research because I work with cancer cell biologists who are in the same building with me. We put together our ideas and we address a number of the real challenges in cancer.
Okay, tell me how you're doing that.
In our lab, we're actually designing a nanoparticle that contains at the very core of it a chemotherapy drug. And around that core, which is a protective shell, we wrap siRNA. siRNA is a molecule that can silence a gene. And then around that we wrap another protective layer and an outer layer. All of these wrappings allow us to design a nanoparticle that can go through the bloodstream and enter the tumor in a very targeted fashion and deliver both of those drugs.
Now, is it important which one gets wrapped around first?
Absolutely. The order is really critical because the order in which we introduce these things determines when they get released. And it's sort of build it from the inside out and then it gets delivered from the outside in. It turns out that the siRNA actually silences a gene that cancer cells, because they undergo all of these genetic mutations, have special to them that allows them to survive in the presence of chemotherapy drugs. So if we understand something about a given cancer, about its genetic code, we can understand what those genes are that defend the cancer cell and keep it from responding to chemo treatment. If we deliver that first, we need a few hours to actually silence that gene mechanism, and then the chemotherapy drug gets delivered. It will get delivered to a cell that can die in the presence of chemo drug.
What kinds of cancer is this useful for?
This is useful for highly aggressive and recurrent tumors. So you wouldn't use it for a cancer that can be addressed using more of our well-known chemotherapy agents. But in some cases, there are patients whose cancers come back. And they come back in this really aggressive form because this is the cancer cell that survived that first attack and has a mechanism for defending itself. The kinds that we're treating or working on right now are high-grade ovarian cancer, which is actually very important to us because it hasn't really seen very much progress. It's always discovered quite late. And also non-small cell lung cancer, that's the most aggressive form of lung cancer. And triple-negative breast cancer are some of the types that we've been looking at.
Paula, thank you very much for stopping by the Hub Culture Pavilion.
Excellent, thank you.
Here in Davos, and I'm Edie Lush.